So in this lecture, we are going to cover adaptive immunology. So adaptive immunity is, again, an organism's specific response to a specific pathogen. And so in the last video, we just covered innate immunity, which is your immune response to a non-specific pathogen, meaning it's innate, it's built into your body, it's present at birth. But in adaptive immunity, this is going to target a specific pathogen. And we're going to talk about how your adaptive immunity is divided into two branches. What is called humoral immunity. Humoral immunity is used to target extracellular pathogens, so meaning that pathogens that are not within your cell. This is mediated by B cells. That's in contrast with our cell-mediated immunity, which is used to target pathogens within the cell. So, for example, a virally infected cell. And so that is within the host cell. Cell-mediated immunity is mediated by our T cells. So we're going to look at our two branches and then talk about how those two branches work. So what I want to do to start out is I want to show you a video that summarizes immunology so that it kind of gives you an idea up front of what we're going to talk about. And then you can go back and review this video later if you want to review all of the topics covered in this video.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then, the helper T cell secretes chemical messengers known as cytokines, that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
So that video was your introduction to acquired or adaptive immunity. And so acquired immunity is, again, adaptive immunity that's acquired only after an immunizing event, such as an infection. And T and B lymphocytes undergo a selective process that prepares them for reacting only to one specific antigen, or what we call an immunogen. When we talk about immunocompetence, this is the ability of the body to react with countless foreign substances. And so somebody who is immunocompetent is able to respond to any number of pathogens. The flip side of that would be if we say that somebody is immunocompromised, right? And we've talked a lot about conditions that make you immunocompromised, but basically it means that the immune system is not functioning properly and it's not able to fight off pathogens. And so that would be the opposite of immunocompetence. So an antigen is a molecule that can be seen and identified by the immune system. So it's something that is recognized by the immune system. If they provoke an immune response, they are referred to as immunogens. And proteins or polysaccharide molecules on or inside the cells and viruses can be potentially immunogens. Any exposed or released protein or polysaccharide is potentially an antigen, even those on our own cells. However, our own antigens usually do not evoke a response from our own immune system. And again, this is an important part of an immune response, is the immune system's ability to differentiate between antigens that should cause a response versus those that do not. And if the immune system does not respond properly and it reacts to our own antigens, that's when we end up with an autoimmune disease. The immune system is overreacting. So there are several important characteristics of specific immunity. And so the first property that's important for acquired immunity is what's called specificity. And specificity is when a response is focused on a single antigen. So antibodies that are produced against the chickenpox virus, for example, the varicella virus, will not react against the measles virus. So the immune response is highly specific. There is also a lot of diversity. And so that's because we make a variety of different leukocytes that can respond to any number of pathogens. And so we have a lot of B cells with unique B cell receptors. We have what's called inducibility, meaning that the immune response is only turned on when triggered. This is used to save resources. The body doesn't want to spend energy to produce a bunch of B cells, for example, that are not needed. And so this system is inducible. It's only turned on when needed. Clonality, so this will generate millions of cells with the same specificity. So once our immune system is initiated, and once we start to recognize a specific pathogen, that particular B cell or that particular T cell is going to undergo what we call clonal expansion. And it's going to produce lots of clones that recognize the same antigen. The immune system also has what we refer to as tolerance, and that is that it does not react with self-antigens. So again, it's not going to target our own cells, but instead target foreign invaders. And then we have the memory component. And this is where lymphocytes have been programmed to recall their first engagement with the invader and rush to the attack once again. And so, for example, if you've had chickenpox, your immune system remembers that. And so if you were to come in contact with the virus again, you are not going to get the chickenpox because you still have that memory component that remembers that antigen. And so if you were to come in contact with it again, your immune system is going to mount a quick response and you're not going to see the symptoms. And so this is why we've seen more recent outbreaks, like the measles outbreaks that we've seen, because if people are not immunized against a particular pathogen, and we'll talk later about herd immunity, but if people are not vaccinated against the measles, whether it's by choice or whether it is because they're children and they're too young age to be immunized, but people who are not immunized don't have that memory. So that if you have a percentage of the population that do not have immunity, right, they've not seen this pathogen before, they don't have that memory component, and they have the potential to spread it to other people. And so that's why with the coronavirus, one of the things that's so important is to achieve a point where we have herd immunity, meaning that the bulk of the population is immune to the coronavirus, and therefore it can't be passed to cause an outbreak, essentially. And so the immune system, one of the important features of the immune system is that memory component, remembering a particular antigen so that the immune system can respond quickly if challenged with that antigen again. So when we talk about acquired immunity, there are 4 main steps in terms of acquired immunity. And so the first step is going to be lymphocyte development and clonal deletion. So making the appropriate lymphocytes and deleting the ones that could be harmful, for example. Presentation of antigens and clonal selection. So recognizing something as foreign, then presenting that antigen and selecting for the appropriate lymphocytes that would recognize that antigen. Challenge of B and T lymphocytes by antigens, and then the T lymphocyte response, which is what we call cell-mediated immunity, and the B lymphocyte response, which is going to include humoral immunity and the production and activities of antibodies. And so we're going to walk through and talk about these different stages and how they are achieved and how they occur. So we have, again, this dual nature of adaptive immunity. This is going to be part of our third line of defense. And our third line of defense can be broken into 2 branches. The first is going to be humoral immunity. And again, that's going to be used to fight invaders outside of our cells. So they're not within our cells, but are outside the cells. So this would include things like bacteria, toxins, etc. Humoral immunity is mediated by B cells. And B cells are produced, they're called B cells because of bursa. They are produced in the bone marrow. And then they mature in the bone marrow, and then they're released to tissues. And we're going to talk about later that those B cells can be responsible to produce antibodies or to make memory B cells. And those antibodies are used to fight pathogens outside of the cells. The other arm of adaptive immunity would be our cell-mediated immunity. And this attacks antigens that are found inside of our cells. So this would include viruses, some fungi, and parasites, things that are found within our cell. Cell-mediated immunity is formed by our T cells, and the tissue where T cells will mature and develop would be in the thymus. And so the thymus are going to produce those T cells, and then those T cells, specifically our cytotoxic T cells, are used to fight pathogens inside the cell. And so we're going to look at more detail of how these 2 branches of immunity occur. So we're going to go through stage 1 and talk about the development of lymphocytes. So when pathogens carrying antigens cross the first line of defense. So again, when we overcome the first line of defense, remember that the first line of defense is barriers to prevent a pathogen from getting in. Once an organism has breached the first line of defense, that is going to activate our second line of defense. And so we talked about that part of our second line of defense is that these resident phagocytes are going to migrate to the site of where the pathogen has entered the body. The tissue macrophages are going to ingest that pathogen and initiate an inflammatory response. So again, that is all part of our second line of defense. Dendritic cells ingest the antigen and then migrate to the nearest lymphoid organ where they process and present antigens to T and B lymphocytes. So now we're starting to see what is referred to as antigen presentation. And now we're starting to cross over into our adaptive immunity, because now we're presenting a specific antigen to our T and our B lymphocytes. And then gamma delta cells can be activated by those PAMPs. Remember that those are molecules on the surface of bacteria, for example, that are conserved among a variety of organisms, and they signal to the immune system that that organism is going to be foreign. And so that is going to turn on and initiate this response. All cells have a variety of different markers on their surface that are used for a variety of purposes. One, they're used for detection. So detecting whether the cells are self or non-self. They have different markers for recognition. So again, is it self or non-self? Remember that when we watched the video of the white blood cell chasing the bacteria, did the white blood cell engulf the red blood cells? And the answer is no. You didn't see the white blood cell doing phagocytosis of the red blood cells, because there are markers on the surface of the red blood cells that are seen as self, but the bacteria has markers that are seen as foreign. Markers also can play a role in cell communication, how cells talk to one another. And so these markers can play an important role in activating different components of immunity, whether it's going to be an antigen that is an immunogen and stimulates an immune response or not. So in order to understand these markers, we have to talk about what are called major histocompatibility complexes. And these are a set of genes that code for human cell markers or receptors. These give rise to a series of glycoproteins that are found on all cells except red blood cells. So every cell in your body except red blood cells have these MHC molecules. And they're also known as the human leukocyte antigen, the HLA system. These markers play a vital role in recognition of self by the immune system and in rejection of foreign tissue. And so if you think about it, if you know anybody who has had an organ transplant, for example, right? If they get an organ transplant, they need to find a match. Somebody whose MHC are close enough to the patient who's receiving the organ, they have to be similar enough so that the immune system doesn't mount an immune response and start to attack this organ that's been transplanted into the body. And so it's important that if you get an organ transplant, that you find a match who has the appropriate MHC. So when we talk about MHC, these can be divided into two major classes. Class 1 MHC, what we call MHC1. MHC1 genes code for markers that appear on all nucleated cells. So again, all nucleated cells, except it's not found on red blood cells, because red blood cells are not nucleated cells. So all the other cells in the body are going to have class 1 genes. These display unique characteristics of self and allow for recognition of self molecules and regulation of immune reactions. And so what that means is that if an antigen is displayed on MHC1, if an antigen is being displayed on MHC1, what that tells the immune system is that cell is infected. It's like telling the immune system, I'm infected, I need to be destroyed. And so MHC1 is going to be used to display peptides from endogenous antigens. meaning from within the cell itself. And that MHC1 is going to be recognized by a molecule called CD8, and CD8 is found on cytotoxic T cells, so TC cells. And so if an antigen is displayed on MHC1, again, it tells the immune system, I'm infected, myself, I'm infected, destroy me. When the antigen is displayed that way, it's going to bind to the cytotoxic T cells. And remember that the cytotoxic T cells are the ones that do the cell-mediated immunity. And so the T cells, the cytotoxic T cells, are going to recognize the antigen displayed that way, and it's going to tell the immune system to kill that cell. So class 1 is I'm infected, destroy me. Class 2 serves a different purpose. So MHC2 codes for immune regulatory markers found on macrophages, dendritic cells, and B cells. And these are involved in the presentation of antigens to the T cells. So the cells that produce these are our antigen-presenting cells, what we call APCs. And when it's presented with MHC2, those antigen presentation cells are telling the immune system, I'm not infected, don't destroy me, I'm just the messenger. When it's displayed on MHC2, it's telling the immune system, this is the antigen you're looking for, but you need to go find it elsewhere. I'm not infected. And so this is going to be recognized, these MHC2 are recognized by CD4, and CD4 is found on T helper cells. T helper cells, you're going to learn in a little bit. These are the commander of the army. When the T helper cell becomes activated, it's going to send out what are called cytokines. These are those chemicals that are going to initiate an immune response. So our antigen-presenting cells present it on MHC2, and that is going to signal to the immune system. It's going to activate the T helper cells, and it says, this is the antigen, go find it elsewhere. And so this is going to be used to display peptides from external antigens, meaning it's not within the cell itself. It's telling the immune system to go find this somewhere else. For me, when I'm trying to remember which CD molecule goes with which MHC, multiply the two together and it always equals 8. So MHC1 goes with CD8 because 1 times 8 is 8. MHC2 goes with CD4 because 2 times 4 is 8. So that for me is how I can keep track of which MHC goes with which CD molecule. And so our CD4 is going to be found in our T helper cells. And again, the T helper cells are going to recognize it by MHC2 because the T helper cells are just there to pass on the signal to other cells. The cytotoxic T cells, the TC cells, those are the ones that are going to destroy self-infected cells, like virally infected cells. And so our TC cells are going to recognize antigens that are displayed on the MHC1. And again, all nucleated cells are going to have the ability to display antigens using MHC1 because all the cells in your body could have the potential to be infected or have a problem. So we have 2 main classes of MHC. One is going to signal that that cell itself is infected, target it for destruction. There's another MHC that can display molecules, display antigens, and it's just there to display the antigens. It's an antigen-presenting cell, but it's telling the immune system, go look elsewhere for this pathogen. So, for our lymphocyte receptors, the major role is to accept or grasp antigens in some form. B cells have receptors that bind to antigens, and our B cells have what is called a B cell receptor. And the B cell receptor is very specific for a particular antigen. T cells have receptors that bind to antigens that have been processed and complexed with the MHC molecules on the presenting cell. So not only does the T cell need to have the receptor that binds to the antigen, but the T cell, again, also has the appropriate CD molecule that is going to recognize the MHC. So the T cell has two bindings that have to occur. It has to have the T cell receptor binding to the antigen, and it has to have the appropriate CD molecule that binds to the appropriate MHC. Receptors have the capacity to respond to nearly an infinite number of different antigens. And that is why we say that we are immunocompetent. We have the ability, our immune system has the ability to respond to almost any pathogen. Now, when challenged with an antigen, both B cells and T cells proliferate, which means that they divide, and differentiate, which means that they become specialized. So they become differentiated to become a specific type. So in the case of a B cell, when they differentiate into two main types of cells, either plasma cells, which produce antibodies, or memory B cells that remember the pathogen. So that's differentiation. They become specialized. So when challenged with antigens, our lymphocytes will become a clone, which is a group of genetically identical cells. And that's because that when that B cell, for example, gets stimulated, it's going to stimulate it to divide, proliferate, which is what's going to create a clone, because it's just going to divide and make a copy. Some clones are going to be memory cells, and these memory cells will ensure future reactiveness against a specific antigen. So that again, if the antigen is encountered, the immune system can mount a very quick response, and the patient is not going to show symptoms of the disease because the immune system is going to fight it off before disease can occur. B cell and T cell responses are going to differ, and we're going to see this in a little bit. So there are three functional main types of T cells. We have our helper T cells, or T helper cells. They're called either one. And our helper T cells are used to activate macrophages, to assist B cell processes, and to activate cytotoxic T cells. You can think of our T helper cells as the commander of the army. They're the one that sends signals to everybody else, activating everybody else. We have our regulatory T cells. These are going to control the T cell response by secreting anti-inflammatory cytokines or preventing proliferation. So this is to help turn off an immune response. We have our cytotoxic T cells. These lead to the destruction of infected host cells and other foreign cells. And so again, this is going to be part of our cell-mediated immunity. T cells secrete cytokines to help destroy pathogens, but they do not produce antibodies. So that's one of the things that makes T cells and B cells different, is that B cells are primarily responsible for antibodies. T cells are not. T cells will release cytokines, but they do not produce antibodies. The antibodies are left up to the B cells. B cells. When activated, B cells divide and give rise to plasma cells. Plasma cells are the ones that release antibodies into the tissue and into the blood. And then the antibodies are going to attach to the antigen for which they are specific, and the antigen is marked for destruction or neutralization. And so in a little bit, we're going to talk about what are the functions of antibodies. How do they work to stimulate an immune response? So this slide is just comparing and contrasting B cells and T cells. B cells mature in the bone marrow. T cells mature in the thymus. In terms of their specific surface molecules, B cells have what are called B cell receptors, and they will produce these immunoglobins. T cells, on the other hand, have what's called a T cell receptor, and it has several CD molecules which recognize MHC. And so if we look at what they bind, so let's write a note on here. Our T cell receptor, it's on. My T cell receptor is going to recognize an antigen, and the CD molecules are going to recognize MHC, the appropriate MHC. In terms of circulation in the blood, B cells are found in low numbers. T cells are found in higher numbers. The receptor for the antigen in the case of a B cell is referred to as a B cell receptor or BCR. And again, that B cell receptor is an immunoglobin. It's essentially an antibody that's linked to the membrane. A T cell has what's called a T cell receptor or TCR. We'll skip the distribution in the lymphatic organs. I'm not going to ask you about that. Require antigen presented with MHC. B cells, no. They don't require MHC presentation, but T cells, yes. And again, that's because the T cells have those CD molecules which recognize the appropriate MHC. Product of antigenic stimulation, B cells are going to produce plasma and memory cells. T cells are going to produce several types of activated T cells as well as memory cells. General functions of B cells, so to produce antibodies, to inactivate, to neutralize, and to target antigens. For the T cells, their function is that these are cells activated to help other immune cells. They're used to suppress or kill abnormal cells, to mediate hypersensitivity, and to synthesize cytokines. And so their functions are going to be distinct. They work together, but they're distinct. So for our T cell, maturation of T cells and development of the receptors is directed by the thymus gland and its hormones. And so again, if we look at the surface of a T cell, it's going to have its T cell receptor. So that's this little green one here. This is our T cell receptor, which recognizes a specific antigen. Also, we are going to have what we call co-receptors. We have CD3. CD3 is a receptor that surrounds the T cell receptor and helps with binding. And so you can see it on either side, and it's going to help with binding. We have CD4 co-receptors. Again, CD4s are going to recognize MHC2. So the CD4 is found on my T helper cell, and its job is to recognize MHC2. CD8 is found on cytotoxic T cells, and it helps to bind MHC1. So these are the different types of receptors or co-receptors that can be found on the surface of our T cell. Our B cell is going to develop in the bone marrow. And so our naive lymphocytes circulate in the blood, homing to specific sites in the lymph nodes, spleen, and other lymphoid tissues, where they adhere to specific binding molecules and come into contact with antigens throughout the life. So notice that on the B cell, if we look at the surface of the B cell, it has what we call our B cell receptor. And our B cell receptor is our immunoglobin. Again, it's essentially an antibody that is embedded in the membrane of the B cell, and it has its antigen binding sites. So it has 2 sites to bind antigens. You'll notice that on the surface of the B cell, it's also going to have MHC2 and MHC1, because all nucleated cells have MHC1. B cells are going to have MHC2 because a B cell can be an antigen-presenting cell. It can present antigens to the T cells, and so it has its MHC2 as well. So by the time B and T cells reach lymphoid tissues, each one is equipped to respond to a single unique antigen. And so the diversity of these antibodies and these receptors is generated by the rearrangement of gene segments that code for antigen receptors on T cells and B cells. And so what is absolutely amazing about the immune response is that we have these genes in our DNA sequence. And in antibody production, for example, there is a type of alternative splicing that's called VDJ rearrangement. And VDJ has to do with this antigen binding site that's found on the antibody. And so we have this gene and it has these different components, but we can have splicing of the mRNA to produce a wide variety of different mRNAs from a single gene. And so notice that this one over here has some particular molecules. This one is different. This one is different. And so every possible recombination occurs, leading to a huge assortment of lymphocytes. So we are able to take a very small number of genes and do this VDJ rearrangement, do this alternative splicing, and cut the mRNA in unique ways to produce an infinite number of different antibodies. And so it's estimated that each human produces antibodies with 10 trillion different specificities. So think about that, 10 trillion different specificities. That's a lot of genetic diversity. And again, it all comes down to this alternative splicing. So we're going to look now at a little bit more information about our B cell receptor, or our immunoglobin. Immunoglobin is referred to as Ig, and these are large glycoprotein molecules that serve as antigen receptors of B cells. When they are secreted from the B cell, we call them antibodies. So when they're on the surface of the B cell, we just simply call them immunoglobin. If they are secreted and released, from the plasma cells, which are a specific type of B cell, then we call them antibodies. If we look at the structure of an immunoglobin, it is a 4-protein chain that forms a Y shape. It has what we call 2 heavy chains and 2 light chains. The heavy chains are called heavy because they have a higher molecular weight. The light chains are a smaller molecular weight. So when we get an antibody, it has 2 identical heavy chains and 2 identical light chains. And those chains are held together by these disulfide bridges, these sulfur-sulfur interactions that hold them together. When we look at the structure of the antibody, we have what's called the antigen binding site. And the antigen binding site is going to be the pocket in the end of the fork of the molecule that is highly variable in shape to fit a wide range of antigens. And so this is where the specificity comes into play, is at this antigen binding site. So this antigen binding site is going to bind to a specific, what we call epitope or antigenic determinant. It's a part of a particular antigen. And so the specificity comes from this antigen binding site. It's going to have a particular structure, which is going to allow it to bind to a particular antigen. When we look at the immunoglobin, we have what's called the variable region, and the variable region includes the antigen binding site. This is the area of extreme versatility from one clone to another, meaning that that's going to make different immunoglobins different from one another. And there is also what is referred to as the constant region. And the constant region is found on both light and heavy chains. And this is the part of the immunoglobin that does not vary greatly from one antibody to another. So we're not going to get into the specifics, but there are a variety of immunoglobins, IgG, IgA, IgM, etc. And for each class of immunoglobins, the constant region is relatively the same, meaning there's not a lot of diversity in that sequence per immunoglobin. So IgG is going to have a very similar constant region. IgM is going to have a very similar constant region. The part that varies between one antibody and another is going to be the variable portion. That's the part that binds to the antigen, and that's the part that's really going to vary. So when we look at when clones are produced, we call this process clonal selection. And so clonal selection is the mechanism by which the exactly correct B cell or T cell is activated by an incoming antigen, meaning that if the B cell or T cell has the appropriate B cell receptor or T cell receptor, that lymphocyte is going to be stimulated to produce more of itself. That's the clonal selection. Lymphocyte specificity is pre-programmed, existing in the genetic makeup before an antigen has ever entered the tissues. So what you have to think about is that this is completely random. It's not like your body comes in contact with the antigen and then makes the appropriate B cell receptor or makes the appropriate T cell receptor. That's not how this works. How it works is in your body, you have a lot of B cells and T cells that have a variety of different receptors. And if that receptor happens to come in contact with its antigen, then that particular lymphocyte is going to be selected for. But again, it's not like it's a conscious decision on the immune response. Those lymphocytes have already been pre-programmed. They already have their genetic makeup. They have already made their B cell or T cell receptor, and so it's pre-existing. Each genetically distinct lymphocyte expresses only a single specificity and can react to that chemical epitope. So even within a particular pathogen, so let's say the flu, for example, there are a variety of antigens that can be present on the flu virus. And just because you make antibodies to one part of the virus, that doesn't mean it's going to respond to another part of the virus. And so for the flu, one of the reasons that you have to get a new vaccine every year is because the flu virus mutates. And if it mutates and it changes its surface molecule, yes, you may have had the flu last year, but the new flu the next year is not the same. It does not have the same antigens, and therefore your memory component is not going to work because your immune system doesn't recognize those new antigens. It's going to have to respond again. And so these receptors are highly specific. They react to a single type of antigen. And so that's going to be an important part. Clonal expansion is the rapid multiplication of B cells or T cells after activation by the antigen. So when that lymphocyte binds to its antigen, then it's going to be stimulated to proliferate, to divide. That's going to be our clonal expansion. That's what's going to lead to those clones that all have the same receptor for that particular antigen. Now, one potentially problematic outcome of random genetic assortment is the development of clones of lymphocytes that have the ability to react to self. So again, those receptors are going to be random, right? It's not anything particular that is being recognized. And so an important part of the immune system, in addition to clonal expansion and clonal selection, is clonal deletion. And this is the process by which any such clones are destroyed during development. So that leads to what's called immune tolerance, so that the immune system doesn't react with self molecules. And so those particular lymphocytes are selected against, and those lymphocytes are going to be destroyed. And so that's a very important process. So you can see that here, this lymphocyte and this lymphocyte, they react with self molecules, and therefore the immune system is going to delete or get rid of those cells that can recognize self, so that we don't end up with an autoimmune disease. So we talked about how our lymphocytes develop. Now we need to talk about how they work. And so the next part of this is looking at stage 2, which is the presentation of the antigens. How does the immune system display the antigens to activate our adaptive immunity, which is going to be our specific branch of our immune system? And so we're going to talk about antigen presentation. And so this little cartoon is showing this phagocyte, and it's doing phagocytosis. It's engulfing this foreign invader. The phagocyte is going to break it down, and then it's going to display the antigen on its surface. And this helper T cell is going to have the appropriate receptor. It's going to bind to the antigen, and it's going to stimulate and activate the T cell. And so that's just a cartoon version of what you're going to see. So in most immune reactions, antigens must be formally presented to lymphocytes by antigen-presenting cells. And so examples of antigen-presenting cells are going to include our macrophages, which are our phagocytes that do phagocytosis. We have B cells that can act as an antigen-presenting cell. And we have our dendritic cell. So what you're seeing here is we have our antigen-presenting cell. And this antigen-presenting cell, let's say this is a macrophage, it's going to undergo phagocytosis, and it's going to engulf this microbe. And it breaks down the microbe, and it's going to display its antigens on MHC2. Because again, my APCs are going to display on MHC2, which is what tells the immune system, this is the antigen you're looking for. Don't kill me. I'm just the messenger. but go find this antigen elsewhere. And so it's going to be put on MHC2. A T helper cell is going to come along, and the T helper cell has to have the appropriate T cell receptor, and the T cell receptor is going to bind to the antigen. It has to have the CD4, and the CD4 is going to bind to the MHC2. We also have the CD3 co-receptor, which just helps with binding. But we need all of this to come together. So the T cell receptor needs to bind to the antigen. The CD4 on the T helper cell has to bind to MHC2, which is going to activate this T helper cell. What you're going to see later is that the macrophage also secretes a cytokine called interleukin 1. Interleukin 1 is going to stimulate or activate the T helper cell. It's going to cause that T helper cell to undergo clonal expansion, right? It's going to help this T helper cell to be activated, to proliferate, to differentiate, etc. What you're going to see later is that when the T helper cell is activated, then it's going to produce a cytokine. And it's going to produce a cytokine called interleukin 2. Interleukin 2, basically, you're going to see, activates everybody else. It activates the antigen-presenting cell. It activates the T helper cell itself. It activates our B cells. It activates our cytotoxic T cells. It basically activates everybody else. And so the purpose of our antigen-presenting cell, though, is to take in the pathogen, break it down, and then present the antigen using MHC2 to help stimulate the T helper cell. Because again, the T helper cell is going to be the commander of the army. It's going to tell everybody else what to go do. So looking at our antigen-presenting cells, we again have our dendritic cells. Dendritic cells have a structure that looks similar to a neuron, for example. It has all of these little extensions. And the purpose of the dendritic cell is to engulf and degrade microbes and display them to the T cells. These cells are found in particular tissues. So it's found in the skin, it's found in the genital tract, it's found in the lymph nodes, the spleen, the thymus, and the blood. And so in these tissues, this is where dendritic cells are going to be found to act as an antigen-presenting cell. Macrophages are going to be activated by cytokines or by the ingestion of antigenic material, meaning they do phagocytosis and they engulf that pathogen, and that stimulates them to become activated. So again, our macrophages perform phagocytosis. They will then migrate to the lymph tissue and present the antigen to the T cell. Again, macrophages will also secrete interleukin 1. Interleukin 1 stimulates inflammation and fever. It also activates T helper cells. And so these are two types of antigen-presenting cells. Now, most antigens must be presented first to T cells, even though they will eventually activate both B and T cell systems. T cell independent antigens are antigens that trigger B cells directly without antigen-presenting cells or T helper cells. So this type of antigen can stimulate B cells without a T helper cell. And when that happens, it provokes a weak immune response and no memory cells generated. And so what you have to think about, and this kind of goes along with what we're thinking about in terms of coronavirus. right? And while we're trying to figure out the appropriate response in terms of the coronavirus, one of the things that's happening right now is antibody testing. And what that could tell us potentially is that if somebody has antibodies in their body against coronavirus, it doesn't mean that they have the infection now, but they might have the memory component. However, presence of antibodies does not necessarily mean that the memory has been stimulated. T cell independent antigens don't cause memory. And so even if you were to detect coronavirus antibodies, that does not necessarily mean that you are immune to the coronavirus. That does not mean that you can't get COVID-19. You could. Research is still trying to determine whether or not that is true, whether or not it initiates the memory component of the immune system. Not all antigens do. Not all antigens will stimulate an immune response that is strong enough to provide memory. So a T cell independent antigen provides a weak immune response and no memory is generated. T cell independent antigens are usually carbohydrates with many repeating and invariable determinant groups. This can include the lipopolysaccharide, the LPS, from the cell wall of E. coli, polysaccharides from the capsule of Streptococcus pneumoniae, or molecules from rabies or Epstein-Barr virus. So these would be examples of T-cell independent antigens. This is able to evoke an immune response without the help of a T helper cell. And so this, again, is going to be a weaker immune response and no memory is generated. So next, we're going to look at stage 3 and 4, looking at B cell activation and humoral immunity. And again, this is used to fight pathogens outside of our cells. So clonal selection is going to differentiate activated B cells into two types: plasma cells, which secrete the antibodies, and the memory cells, which recognize pathogens from previous encounters. And so what you're seeing is in this little cartoon, this B cell is going to find an antigen that matches its receptor. And when it finds that particular antigen, what you're going to see, and this is not shown in this image, is that that pathogen is going to be ingested by the B cell. And then the B cell is going to be an antigen-presenting cell. And it's going to present the antigen, and it's going to be activated by the T helper cell. When the T helper cell activates the B cell, that is going to stimulate the B cell to proliferate, meaning make more of itself, so clonal selection, and to differentiate. And so it's going to differentiate into two types: plasma cells, which are going to secrete antibodies, and memory B cells, which remember the pathogen. And so when the pathogen is targeted by the antibodies, for example, that is going to stimulate phagocytes to destroy that pathogen. So it's fighting bacteria that are outside of our cells. The memory cells will remember that pathogen so that if you come in contact with it again, it's going to respond much faster. And so one of the things that you're looking at on this bacterial cell down here is that this is a bacterial cell and it has antigens. And these antigens are components that are found on the cell wall. Within each antigen is what is called an epitope. An epitope is an antigenic determinant. Antibodies bind to one particular epitope. Notice that it doesn't bind to the whole antigen. It only recognizes one part of the antigen. So it only recognizes one epitope. So antibody A recognizes that epitope. Antibody B recognizes a different epitope, etc. So this diagram is used to help you with question 168 on your study guide. And it asks you to describe clonal selection of B cells. So let me walk you through this. So we have our B cell, and our B cell has its B cell receptor. And the B cell receptor, remember, is an immunoglobin, and it has a binding site for a particular antigen. So when this B cell encounters its antigen, that B cell receptor and the antigen are going to be brought into the B cell. The B cell is going to destroy it. It's going to break it down. And then it's going to present its antigen using MHC2, right? So notice that it's presenting the antigen on MHC2. And that, again, is telling the immune system, don't destroy me, go find this antigen elsewhere. And so what comes along is a T helper cell comes along, and the T helper cell has a T cell receptor that binds antigen. So it binds to the antigen that is being displayed by the B cell. The T helper cell also has CD4, which binds MHC2. So the B cell is displaying its antigen on MHC2. So my T helper cell comes along, and the T helper cell has a T cell receptor, and the T cell receptor is going to bind to the antigen. The CD4 is going to, it's not really shown here. That would be like this other part. The CD4 is going to recognize that MHC2. And when both of those occur, my T helper cell is going to secrete cytokines like interleukin 2, like B cell growth factor, other cytokines that we'll talk about in a little bit. But my T helper cell is going to then activate this B cell. And that is going to select for this B cell. That B cell is going to undergo clonal expansion. It's going to be stimulated to divide, right, to produce more of itself, and to differentiate, to become specialized. And one of the cells that it can become specialized to is to become a plasma cell. And that plasma cell is going to secrete antibodies against the particular pathogen, because notice it has the appropriate immunoglobin on its surface. So when it gets stimulated to become a plasma cell, that plasma cell is going to produce these antibodies, and those antibodies are going to go look for the pathogen outside of the cell. So for question 168 on your study guide, this is the diagram that it's referring to. It's referring to this clonal selection of B cells and how does this occur. So this is what we're looking for. So this is just showing it in a little more detail. So again, we have our B cell, and my B cell has my immunoglobin, my B cell receptor, that is going to bind to the appropriate microbe, meaning it's going to bind to the antigen. When it binds to that microbe or that antigen, it's going to internalize that antigen. It's going to break down the microbe, and it's going to display the antigen using MHC2. So again, my B cell is going to display it on MHC2. My T helper cell is going to come in. It has its T cell receptor. T cell receptor recognizes the antigen. The CD4 is going to recognize the MHC2. And when that occurs, now my T helper cell is going to produce cytokines that activate the B cell. And so you can see that it says interleukins are going to be produced, B cell growth factors are going to be produced, and that is going to lead to the activation of that B cell. And that B cell is going to proliferate, it's going to grow, and it's going to become differentiated into regulatory B cells, which we're not going to talk much about, memory B cells, which remember, and the plasma cells. And the plasma cells are going to undergo clonal expansion, and they're going to produce a lot of antibodies that can fight off that infection. This is why if you've not come in contact with a pathogen before, right? Let's say you haven't been exposed to the chickenpox virus, the varicella virus, and you've not been immunized. If you come in contact with the virus, it's going to take your immune system some time to mount this response. It's going to take it time to produce antibodies to fight off that infection because it's never seen that before. However, if you have either already had the chickenpox or you've been exposed to the antigen artificially through immunization, that means that you have those memory cells, and those memory cells remember the antigen. And therefore, if you were to come in contact with the chickenpox virus, your body is going to respond very quickly because those memory cells are already there and they remember that antigen. So what is the purpose of the antibody? What does the antibody even do to help fight off foreign invaders? And so there are five main purposes of the antibody. There are 5 things that antibodies do to protect the body. The first is that these antibodies do agglutination. We learned about agglutination when we talked about blood typing, right? Because antibodies are Y-shaped structures, and the antibodies have 2 binding sites. 2 binding sites for 2 antigens. And so what that does is notice that because it has 2 binding sites, it causes this clumping or this agglutination of bacteria in addition to red blood cells, right? Foreign red blood cells are like a bacteria in essence. It's detected as being foreign, which is why antibodies react. So the antibodies do this agglutination, and the purpose of doing agglutination and to cause this clumping is that it reduces the number of infectious units to be dealt with. It's like holding it all together to make it easier to round up rather than them all being spread out. The next thing that it does is something called opsonization. I talked about opsonization when I also talked about complement. Remember that one of the functions of the complement system is that the complement proteins can do opsonization as well. It can mark the microbe, it can attach to the microbe and target it for destruction to the phagocytes. Same thing with the antibodies. The antibodies can coat the microbe and enhances phagocytosis. It's marking that pathogen for destruction. We have neutralization. So if you're thinking of viruses, for example, viruses have to get into a cell in order to cause an infection because viruses are obligate intracellular parasites. If the antibody coats the virus, that is going to block the virus's ability to bind to the appropriate receptor on the, it's going to block the virus from binding to the host cell. And if the virus can't get in, it's not going to cause infection. Same thing with the toxin. A bacteria could produce a toxin, and if antibodies are produced and they bind that toxin, it's going to prevent that toxin from being able to bind to its target site. And so this is called neutralization. It's basically preventing the pathogen or the toxin from getting to where it needs to go to cause the infection. So it's neutralizing the pathogens that are outside the cells. We have what's called antibody-dependent cell-mediated cytotoxicity. And so antibodies are going to attach to the target cells and cause destruction by macrophages, eosinophils, and natural killer cells. So notice if we have this large target cell, right, and this large target cell can't undergo phagocytosis, that's not a possibility because that cell is too large for the white blood cell to engulf. Instead, the immune system is going to kill it chemically. And so the antibody is going to bind to that large pathogen, that large parasite, and it's going to stimulate other immune cells to kill chemically. So you can see that this eosinophil is producing perforin and lytic enzymes that help break down, let's say, a helminth infection, a worm infection, for example. And then lastly, antibodies can activate complement protein. And so remember that if complement is activated, it causes inflammation as well as cell lysis, because complement is going to activate the membrane attack complex. And so antibodies have binding sites for the complement. It's going to bind at its constant region. And so when the complement binds to the antibodies, it's then going to cause that complement system activation. And that's going to lead to inflammation as well as cytolysis, lysis of the cell. And so these are the variety of ways that antibodies can function to fight off a foreign invader. It plays a variety of roles to help fight off an infection. So this is just putting what I just talked about in words. So the functions of the antibodies: neutralization. It's going to bind to toxins or viruses, which make them no longer able to attach to the cell. It plays a role in complement fixation because it's going to bind to the antigen. The portion of the antibody changes shape, exposing complement binding sites. The complement binds, and then it leads to inflammation and cell lysis. Opsonization, again, the antibodies are going to attach. This is going to enhance phagocytosis. So again, it's marking that pathogen for destruction. We have agglutination and precipitation. So you get these large complex or these clumping of antigenic cells. For example, again, incompatible blood types. It does agglutination. And the purpose of this is that it reduces the number of infectious units to be eliminated. So it brings them together and makes it easier to eliminate them. And then we have our antibody-dependent cell-mediated cytotoxicity. And this is where the antibodies attach to the parasite, which then attracts macrophages and eosinophils and natural killer cells, which are going to kill that large parasite chemically because it's not able to do phagocytosis. And so the next part, we're going to talk about stage 3 and 4, but talk this time about T cell activation, what is called cell-mediated immunity. So now we're not looking at humoral immunity and pathogens outside of the cells, but now look at what happens when the pathogen is inside of the cells. So like a virally infected cell, for example. So T cells are used to combat intracellular pathogens. And again, they're going to activate the cell-mediated immunity. These recognize antigenic peptides that are processed by phagocytic cells. So again, our antigen-presenting cells are going to present the antigen, and the T cell is going to have the appropriate receptor. T cells mature in the thymus, and thymic selection eliminates immature T cells. T cells are going to migrate from the thymus to the lymphoid tissues, and T cells will attach to antigens via T cell receptors, what we call TCRs. And so T cell receptors on the T cell surface contact the antigens, causing the T cells to secrete cytokines instead of antibodies. So again, our T cells are going to use cytokines, our B cells are going to produce antibodies. And so this little animation here is showing B cells attack invader outside of the cells, and the T helpers, or the cytotoxic T cells, attack invaders inside the cells. And so they work together, but they each have their own role. And so you're seeing, again, this B cell acting as an antigen-presenting cell. It's presenting its antigen using MHC2. This helper T cell is going to have CD4, which recognizes MHC2. It has its T cell receptor, which recognizes the antigen. And when it binds, the T helper cell is going to secrete cytokines to activate that B cell. So we're going to talk about types of T cells. So the first T cell that we're going to talk about is going to be the helper T cell. So our T helper cell. So it's written with a T subscript H. T helper cells, again, are going to contain CD4. So they're what we call CD4 positive. And our helper T cells are our commanders of the entire system. These are the commanders of the army. They're telling all the other immune cells what to do. So T cell receptors on the T helper cell recognize and bind to antigens that are being displayed by MHC2 on the antigen-presenting cell or the B cell. So again, there's two things that have to happen for our T helper cell. The T cell receptor has to recognize the appropriate antigen, and the CD4 has to recognize the MHC2. So two things have to happen for my T helper cells. The antigen-presenting cell, or the T helper cell, secretes a co-stimulatory molecule which activates the T helper cell. So again, my T helper cell might produce interleukin 2, and my macrophage might secrete interleukin 1. And so we have these variety of cytokines that get produced that stimulate different cells. And so you're going to see that T helper cells produce various cytokines. It produces a wide number of cytokines. So cytotoxic T cells, TC cells, these are CD8 positive. They have CD8. And remember that CD8 is going to recognize MHC1. So what you're looking at here is that we have this infected host cell. And let's say that this infected host cell, let's say it's a epithelial cell in the respiratory system that is infected by a rhinovirus. Well, this virally infected host cell is going to display the antigen using MHC1. Remember, MHC1 is self. It's basically telling the immune system, I am infected. You need to destroy me. And so the cytotoxic T cells are activated into what are called cytotoxic T lymphocytes, or CTLs, with the help of T helper cells and co-stimulatory signals. So basically those TC cells become activated and they become the cytotoxic T lymphocytes. These are the ones that are active and they're able to carry out cell-mediated immunity. CTLs recognize and kill self cells that are altered by infections. And so again, this infected cell, this infected host cell is going to display its antigen on MHC1. And this cytotoxic T lymphocyte, the CTL, is going to recognize the antigen because it has its T cell receptor. So the T cell receptor is going to recognize the antigen, and it has its CD8, which is going to recognize the MHC1. And so that MHC1 is going to signal to the T cell, the cytotoxic T cell, to basically commit suicide, essentially. So what's going to happen is when this binding occurs, the cytotoxic T lymphocyte is going to release what is called perforin and granzyme. Perforin is going to perforate or put holes in the membrane of the infected host cell, and the granzyme is going to induce apoptosis, which is programmed cell death in the infected cells. So this infected host cell, when it's being bombarded with perforin and granzyme, it's going to cause that infected host cell to undergo the apoptosis. And so this is used for virally infected cells, some tumor cells, so that your immune system can recognize those tumor cells as being different than your normal cells, as well as transplanted cells. Because again, if your immune system recognizes something as foreign, it's going to attack it. And so my cytotoxic T cells are used to fight pathogens inside the cell. They're going to cause those infected cells to commit suicide. So these next two slides are looking at the T cell coordinated immune response. And this is what you're going to use for question 176 on your study guide. So let me talk about how the T cell response occurs. We talked about the B cell response. Now we need to talk about the T cell response. So what we have first is our antigen-presenting cell. So our macrophage or our dendritic cell. This picture is showing a dendritic cell, but the same thing occurs for a macrophage. The macrophage is going to undergo phagocytosis. It's going to engulf this pathogen. It's going to degrade the pathogen. It's going to break it down. And then it's going to display its antigens using MHC2. So first thing that's going to happen is that the antigen-presenting cell is going to process the antigen and then display it using MHC2. T helper cells have a T cell receptor. And again, that T cell receptor is specific for the antigen. And the T helper cell has CD4, which binds to the MHC2. So two bindings have to occur for this response to occur. If we're talking about a macrophage can secrete interleukin 1. Interleukin 1 is going to stimulate the T helper cell. So our macrophage is going to stimulate my T helper cell. Now, as a result, so when this macrophage is going to secrete the T helper cell, when the T helper cell becomes activated, that T helper cell is going to secrete interleukin 2. Interleukin 2 is going to activate itself. So it's going to auto-stimulate itself to divide and differentiate. It's going to stimulate B cells, and then those B cells can carry out humoral immunity. Interleukin 2 is going to activate my cytotoxic T cells, and that's going to lead to cell-mediated immunity. And so your T cell response basically is going to help activate everybody. Now, not all B cell responses involve T cells, right? Because we talked about T cell independent antigens, but usually they do. And so the B cells become activated by the T helper cells, and that helps stimulate humoral immunity. And that's going to produce the antibodies, and it's going to produce the memory cells, which is what gives you that memory component. And so the T helper cell, again, is the commander of the army. It's stimulating everybody. And it does so typically by secreting that interleukin 2. So the way that I remember this is IL-1, interleukin 1, comes before IL-2. So the APC is going to be the one that's producing the IL-1. The T helper cell then produces the IL-2, which stimulates itself, and it stimulates everybody else. So other types of T cells involve memory T cells. These are going to recognize a foreign antigen, and it results in a faster response to a subsequent exposure. We have our regulatory T cells, our Treg cells, and these are used to suppress T cells against self and to protect intestinal bacteria required for digestion, as well as protect a fetus. So these are our regulatory T cells that mediate hypersensitivity so that we don't attack our own cells, for example. So the next slide is looking at the types of cytokines. So what you're going to see is the only one of these cytokines that is not produced by T helper cells is the interleukin 1. Interleukin 1 is secreted by the macrophages, and that is going to activate the T helper cells. The T helper cells then secrete interleukin 2. And again, interleukin 2 is going to activate everybody else. IL-1 also can initiate a fever, can generate an inflammatory response. So that's interleukin 1. It's the only one of these cytokines that we're going to talk about that's not produced by T helper cells. It is going to target T helper cells, but it's not produced by T helper cells. Interleukin 2 is secreted by T helper cells, and it triggers T cell division. So it's going to activate T cells to proliferate. It stimulates T helper cells and cytotoxic T cells, and it activates and helps proliferate for natural killer cells, B cells, and T cells. So again, interleukin 2 basically activates everybody. It basically is going to turn on an immune response. It's going to activate humoral immunity. It's going to activate cell-mediated immunity. And it's going to basically turn on the immune response for those cells that have the appropriate receptor. Now, we have some specific types of cytokines as well. My B cell growth factor, or BCGF, it's also referred to as interleukin 4. B cell growth factor, again, is produced by T helper cells. And like the name suggests, B cell growth factor, it helps B cells to grow and divide. So this helps B cells prepare for mitosis, which is cell division, and clonal expansion. So it's stimulating the B cells to divide. We have B cell differentiation factor, BCDF. The differentiation factor is, again, produced by T helper cells. But now, instead of stimulating B cells to grow, this chemical, this cytokine, stimulates B cells to differentiate, to become specialized, to be either plasma cells or memory B cells. And then the last one is going to be our interferon. Interferon is produced by T helper cells and other cells. You might recall hearing about interferons when I talked about innate immunity. And that's because virally infected cells are going to produce interferon, and that interferon is going to be released, And adjacent cells are going to take up that interferon. And then those neighboring cells are going to start to produce antiviral proteins to protect themselves from becoming infected by the virus as well. So the interferons are used to magnify an immune response and to block viral replication. So again, to turn on production of antiviral proteins in cells that have not yet been infected. So next, we're going to talk about the dynamics of antibody production. And so the first time that you are exposed to an antigen, that is going to be your primary response. And so this, again, is when the antigen is first recognized by the B cells. And so in terms of the immunoglobins or the antibodies that are going to be produced, IgM, which is a type of immunoglobin, is produced first, followed by IgG second. And when we have this primary response, we get these memory cells that persist for months to years. Depends on the antigen. Certain memory cells persist longer than others. Now, a secondary response is any subsequent exposure to that same antigen. And so if you come in contact with the antigen again, those memory B cells that are still there can produce plasma cells, and those plasma cells will make those antibodies very rapidly so that you don't see symptoms of the disease. IgM is produced fewer and shorter, but the IgG is produced sooner and larger quantities. And so it's going to help to fight off the infection before you see symptoms of the disease. So this is looking at the dynamics of antibody production. So we're looking at the antibody concentration over time. And so if you're looking at your first exposure to antigen X, notice that it takes a while, maybe a week or so, to start produce antibodies to mount the immune response. And then once we have fought off the foreign invader, the concentration of the antibodies is going to decrease, but those memory cells are going to persist. And so if you are exposed again, you get a second exposure to this antigen. Notice that the immune response happens much faster and the amount of antibodies produced is much, much higher, and therefore you're not going to see the symptoms of the disease. This is also why for an allergic reaction, sometimes the first response, the first time you've come in contact with the antigen, it might not be as severe. However, if you were to, let's say, eat peanuts again, you have those memory cells and the response is going to be much greater the second time, and you might end up with anaphylactic shock, for example. Notice that if you come in contact with a new antigen, right? So antigen Y, again, it's going to take time to produce antibodies to antigen Y. It's not going to have the same peak as a secondary response would be to antigen X, because each antigen has to have its own response. It has a primary response and then a secondary response. And so the last part of this lecture is going to be looking at active versus passive immunity. So when we talk about active immunity means that you actively stimulate an immune response. And what that means is that you are exposed to the antigen. And when you're exposed to the antigen, it mounts an immune response. And when your body mounts an immune response, that can lead to that memory. This active immunity takes time to develop, but can last a long time. And if we look at active immunity, it can be broken down into two categories. We can have natural active immunity. Again, we're still talking about exposure to the antigen, and a natural exposure to the antigen would be infection, right? So you naturally come in contact with the antigen through infection. We could also have artificial antigen exposure, immunization. You are immunized, you are injected with a weakened or an attenuated antigen that is not going to cause disease, but by being exposed to that antigen, it's going to elicit an immune response, which generates that memory. And so that's how these immunizations are able to protect you. Because if you come in contact with an antigen and you mount an immune response and you get that memory component, that helps protect you. So those of you that are younger than I am, you have probably been vaccinated against the chickenpox virus. And so as a result, you were exposed to the antigen without ever getting the chickenpox virus. And therefore, that will be advantageous because you're not going to have chickenpox that's latent in the neurons, which can become reactivated later on. Those of us like me who have been exposed to chickenpox because there was no vaccine when I was a kid, parents purposely infected you with chickenpox when you were little because getting chickenpox as a young kid was much better than getting it older. And so parents would, in some cases, purposely have their kids be around somebody else that has chickenpox so that they would get it early on rather than later in life. And if you are like me and you have been infected with the chickenpox virus, you have latent chickenpox, meaning that it is still in the body. It's never fully eradicated. And under certain conditions, it can become activated. And later in life, you can develop what is called shingles. And so that was discussed in the virology lecture. And so those of you that have been immunized are lucky because you don't have the active virus in your body that is laying latent. You have been protected artificially. Passive immunity. Passive immunity is where antibodies are transferred. No immune response occurs by the recipient. And so if the body does not mount the immune response itself, there is no memory created. This has short-term effects, but the effects are very rapid. And so in terms of passive immunity, we can have natural passive. An example of antibody transfer that happens naturally is from mom to baby via the placenta when the baby is in utero, or breast milk once the baby is born. And so that's why you hear people say, breast milk is good for babies. Well, it is good because any antibodies that the mom produce get passed to baby. And those antibodies help protect the baby. Now, it doesn't help the baby to become immune to that antigen, but it's going to it's going to fight off the infection without the baby's immune system having to produce those antibodies, because babies' immune systems are not functioning properly and they're not able to function in the way that an adult immune system would be. We also have artificial passive, meaning that we introduce antibodies artificially. So examples of this would be antiserums or antitoxins, so to help neutralize toxins that might be in the body. Or we talked about for blood typing, RhoGAM, right? And so if a mom has Rh incompatibility with the baby, mom is Rh negative and baby is Rh positive. For the baby that is Rh positive, when the mom goes into labor and the placenta detaches, we don't want baby's Rh positive blood to interact with mom's, so that mom produces those anti-Rh antibodies. So instead, during delivery, artificially, we inject in those anti-Rh antibodies, and those anti-Rh antibodies, that RhoGAM, is going to neutralize the baby's blood without the mom's body from producing the antibodies and generating that memory. So that's an example of an artificial passive immunity. And then lastly, we have the herd immunity, and that is the presence of immunity in most of the population. And so in that epidemiology video that I showed you, it talked about herd immunity, and we talked about that for different diseases. They have something known as R0, which is how many people are likely to be infected by a single person. And so for coronavirus, for example, it has an R0 of between 2 and 3. And that if we are using an R0 of 2 to 3, in order for herd immunity to protect people so that we don't have an outbreak, meaning we keep the number of cases constant, in order to have herd immunity, at least 50 to 66%, so half to 2/3 of our population, has to have been exposed to the coronavirus, whether that ends up being naturally by contracting the disease, or whether that ends up being artificially when a vaccine is developed. But we need at least, on estimates, 66% of the population to be protected in order for us not to have this increase in the number of cases. And so that's our herd immunity. And so the presence of immunity in most of the population, and that's going to prevent the spread of an infection. And so again, that's why we saw the measles outbreak, because of the anti-vaccine movement that happened, because of an article that was published by Andrew Wakefield, where he claimed that vaccines cause autism. However, it was discovered that those claims were fraudulent. He basically made them up. And so, but unfortunately, that led to a big anti-vaccine movement. A bunch of people stopped vaccinating their kids against the measles, and that decreased that herd immunity, so that now we can get the spread and the outbreak of measles, which had almost been eradicated in the US. We didn't really see cases of the measles because most everybody was protected and you couldn't get an outbreak. But once we lose that herd immunity, once people stop vaccinating, then when we lose that herd immunity, then we can have these outbreaks again. And so that's why you can see these outbreaks of diseases that didn't used to be around before. Measles was pretty much gone until we had this big anti-vaccine movement. And so that's for the herd immunity. So this is just breaking it down for active versus passive immunity. So again, if we're looking at adaptive immunity, we can have naturally acquired. Naturally active means that you come in contact with the antigens naturally. So that happens through infection, and that stimulates an immune response. The body produces antibodies, and it produces specialized lymphocytes, and it generates that memory component. We can have naturally acquired passive. Again, passive means that you're not coming in contact with the antigens, but you are getting the antibodies transferred. And again, this can happen from mother to child, either via the placenta for the fetus, so when the baby is in utero, or to an infant via breast milk. We can have artificially acquired adaptive immunity. We can have artificially acquired active immunity, and that is where the antigens are introduced in vaccines, right? And when the antigen is introduced artificially, it's bringing in the antigen, which is going to stimulate an immune response. And it's, again, going to generate that memory component. We can have artificially acquired passive. And this is where these preformed antibodies are injected into the body. Another good example of passive artificial would actually be what they're doing for COVID-19 cases. They are trying to find people who have recently had COVID-19 and they have a high proportion of these COVID-19, these coronavirus antibodies in their blood, and then take that plasma. People who have had COVID-19 are donating their plasma, which has a lot of these COVID-19 antibodies, and they're then giving it to patients who are really sick with the disease to try and help them to fight off the infection. Because again, it takes time to mount an immune response. But if they inject them with the antibodies against that coronavirus, that will help fight the infection faster. And so that's another example of artificially acquired passive. So, question for you. Maturation of T cells takes place in the red bone marrow, yellow liver, green thymus, blue spleen. So take a minute, pause, think of your answer. When you're ready, push play. Maturation of T cells takes place in the thymus, right? T cells, thymus. And so this concludes our lecture on adaptive immunity.